Awesome, thanks. Hold on, I gotta click that little bar that says it's being recorded. I was gonna take a minute because I have your CVs, Lauren and Maria, but I think what we'll do is just I'll let you introduce yourself because you can do that better than I can. And so I'll go first and then I'll hand it off. And so I don't think Mariana said it, but you all know why you're here. We're talking about all the regulatory things that that go into our job. Which is faculty is more paperwork than anything else. My name is Nathan Tykocki. I'm an associate professor of pharmacology and toxicology at Michigan State University. I've been CAIRIBU adjacent for an unbelievably long time, but he also used to be part of a P20 center that with Sarah Bartoloni, who's here on this call, and a lot of the rest of you, and some collaborations with Wisconsin and so on. So Lauren, I'll hand it off to you.
I'm Lauren Baker. I'm a new assistant professor in the Department of Animal and Dairy Sciences at University of Wisconsin-Madison. Most of my research has been in orthopedics, and then I started working in urology through the K-12 program. Actually, I jumped onto the K-12 and switched my research to urology. Most of my work focuses in using companion animal models to understand complex genetic diseases that could also affect people and dogs as well, mostly dogs. Yeah.
Okay, Maria, all you.
Hi, everybody. I'm Maria Hadjifrangiskou. I'm professor of microbiology and immunology at Vanderbilt University Medical Center, and my lab studies the pathogenesis of urinary tract infections. And lately, we have gained an interest in understanding urinary microbiome pathogen metabolic interactions. I've also been CAIRIBU adjacent for quite a while. I can't believe it's like almost 6 years now. We got a P20 to start a microbial biorepository, and that opened the wonderful scientific floodgates that is CAIRIBU, and I'm excited to be here today.
Awesome. Thank you, Maria. And so I just give you guys a little bit of talk about the format because we just kind of figured it out about 10 minutes ago. As you can see, we have pretty much the gamut of academic career stage, from early career to mid-career to established investigator. And so we're just kind of all going to work together to go over some of these regulatory things, specifically focusing on IRB and IACUC approval, which, as you all know, are things that none of us can do our jobs without. And so I know I'm a little more off the cuff with what I have to say about IACUC and so on. So Lauren put together some slides with IRB, so we're going to let her get started there, and we'll fill in throughout. I know that there's a Q&A at the end, but if it's all right with you, Lauren and Maria, I would just assume if people had questions, we just field them as we go. Sound good?
Yeah, absolutely. That's really fun.
Cool. All right, Lauren, all yours.
All right. Let's see. I would like to share this guy. All right. Are you seeing my slides or the?
Yes, slides. I see them.
Okay, and not the presenter view thing?
Not
Okay,
the presenter
cool.
view.
Okay, great. All right. So I wanted to start this by saying that I'm very new to this. And so this is really about my experience with mostly IRB. So even though I am a veterinarian and I do a lot of, mostly, as I just said, companion animal model research, part of that is comparative research. And so I have a comparative human study. And so over the past couple of years, I had to get IRB approval, and I've been working through a human study. I don't have an IACUC yet, so I can't speak to that, but glad other people can. And yeah, so this is about my experience and all the little mistakes that I made along the way that hopefully people can benefit from. So the first thing that I did wrong was the timeline of this. So just assume the first time you go to get approval that it's going to take a lot longer than you think. I have a pretty simple protocol. I only wanted to get a blood sample and a urine sample from people that were mostly healthy. So I thought it was very simple, but it still took 3 rounds of review before it got approved. So for me, I submitted in March and it was finally approved in May. But there was lots of other stuff that, made this even longer. So just expect it's going to take a while, budget extra time to do this. But the first thing you need to do is find someone that can help you. So when I first started this, I was a scientist in the vet school, and they did not have staff for this. It was sort of everybody kind of had their own way of getting through this. Some people had staff, some people didn't. But I didn't personally. But now in my current department, I do have staff. So look to your staff if they are able to help you. Definitely use those people. Other options, formal mentor, a colleague, staff member, we all have to do it. And so there are lots of knowledgeable people around you that can help you through the process. Some help that I had is one of my mentors shared their protocols and related materials. So that helped me to get a starting point. What do these things have to look like? What does the language have to look like for this? Then you start writing your protocol, right? Use your grants as a guide, go back through, make sure you're very clear about all your numbers and definitions and tools and all the materials you're going to use. All those things are things that your IRB reviewer are going to flag, and that's just another round of approval and rewriting that you have to wait for meetings for them to do that. So try to be upfront, be as specific as you need to be upfront. What is your definition of a healthy person? What is your patient definition? Your inclusion and exclusion criteria should be in a bulleted list, and things that you may not think about, like, are you going to allow pregnant participants? Are you going, is the possibility that a child, someone under age of 18 could be recruited? All of these things are things that they're going to ask about that you'll have to add in if you didn't think about it up front. So they're going to want details on how many visits, what will each visit be? What are you collecting? So all this stuff just has to be very specific. And keep in mind that you also have lots of other supporting information that will go along with your protocol. So your study team, everyone on your study team, so people that are going to be recruiting patients with you, students in your lab, anybody like that, they all have to have the appropriate training, or your protocol, they won't be able to be added to the protocol for approval. So make sure they're doing that ahead of time. It's the first thing I do when someone enters my lab, is everybody gets the training they need, and they go on the protocols before they can touch anything. If you're going to be recruiting patients through phone or email, they're going to want a script or an actual draft of the email that you plan to send out for approval. So be prepared for that. This is all the kind of stuff that made this take a lot longer for me than I expected, because all of this extra thing, I didn't know they needed the email that I wanted to send out, or that they would want me to draft a script. And have your consent form ready to submit alongside the protocol. Remember to simplify any scientific jargon, because they will send it back if it's too sciencey for the average person to understand. And that can be sometimes hard to meet if you're not used to having to do that. So then when you submit, you're waiting for review. Before you submit, ask a mentor or a colleague, that person that's helping you, to review it, to catch any mistakes. There's lots of jargony stuff throughout all of these forms. And you know, you may check a box that you didn't mean to check, and you need to add all these explanations and things like that, but maybe you didn't need to do that, things like that. Remember, you're going to have information about your funding that you have on the project, feasibility statements that may need to be signed by chair. So again, all this stuff that goes along with it, and assume it will be sent back with edits. So once you get it back, you're going to have lots of comments from your IRB reviewer. And I would just say, take a deep breath, because it's their job to be picky. I know it can vary by the person who gets your protocol. I think I was told mine was maybe a little bit pickier than average, but it can be like, oh my goodness, it's definitely not what I meant. And it can get a little frustrating when you're first reading through, but just kind of go through, make sure you're just meeting all of their concerns, resubmit, rinse and repeat until they approve it. And then, yay, you finally do get approval. That may not mean you can start your study right away. So you have approval from the IRB to start your study, but I just wanted to mention that one of the things that we went through is that once we had IRB approval, we actually couldn't start the study because we needed separate approval from the hospital to get into their clinical trial system, and all of that. So that's just another kind of layer of regulation that you need to think about, or you should have thinking about ahead of time, because I think so when I submitted the IRB approval in March, we weren't actually able to start the study until September. So just, that was all the little hiccups we had along the way to getting this going. So be prepared for delays. Definitely don't wait on some of these things. So after you have approval, you can amend it, and you need to amend it if anything changes. The most common reason would be personnel changes, people coming and going from your lab and things like that. Other reasons, if you need to collect more data than you initially planned to, or a different sample type or more sample, you need to submit an amendment to stay good with the IRB. You don't want to end up in a situation where you're doing something that wasn't necessarily approved on your IRB. So just make sure you're doing that ahead of time. And And that amendment may or may not require re-review, but it can be expedited. So hopefully won't slow down your study very much. Definitely something that you should be doing the whole time. And finally, I want to do a quick little bit about reportable events because I got to experience this myself as well. Because mistakes happen. In my case, what happened is that some samples were mishandled, and I didn't realize that was a reportable event, but it was. And so we had to submit a reportable event to the IRB about exactly what happened, how many samples were affected, and what we were planning to do about it. And then they approved that plan and it was fine. But at the moment, it was like, oh no, I had to submit a reportable event. You think it only happens if you made some huge, terrible mistake. But the reality is that mistakes happen and it was fine. And so I can say that this is just something that you may need to do. And so just my final words from my experience is remember that the staff reviewer is not your enemy. They're here to keep you safe. They're here to keep the university safe, and make sure that everybody is following those federal regulations. If you can, get to know members of your IRB staff. IACUC, I've mentioned I don't have an IACUC protocol yet, but I am currently working with one of the vets from our IACUC to make sure that the protocol that I'm writing will be written the way that it needs to be for what I actually want to do. And use the experience of your colleagues to help you through this process and report everything so that in case something goes wrong, you do have everything available to make sure that you're can quickly and easily deal with that. And then so that's all I've got for you guys.
Awesome. I have very little to add because my one IRB that I have is not remotely this complex. But I think one thing I wanted to ask you to address, Lauren or Maria as well, when it comes to IRB, when do you start? Like, do you wait until you have funding? I know some people's universities are this is a weird thing. You sometimes need an IRB to be able to get the funding, but they want to have the funding before they give you the IRB. And so how do you usually work this, Lauren?
I can only speak from my experience in that I already had the K-12, and the first half of my project was really using dog information and dog data. That was existing data, so I didn't need the IACUC. But so I waited until I was about ready to start the human study. So I wasn't too tripped up by the delays that we had because I already had plenty already happening, and it worked out totally fine. But so that's my only experience. I don't know. Maria has something else to add.
No, it's similar. We write it as we need. We write our IRBs as we need them. If we have a project with a collaborator that can provide us patient samples for a specific study, we actually are proactive and write the IRB. And usually that makes it much more easy when you actually go to the NIH if it's existing. And it just makes it a lot smoother. One of the things that I wanted to add, because we do this a lot, when you're ready to write your first R, beware that there is a section that's human samples or material that are not considered human subjects, but they require a small write-up. For example, we use derivatives of clinical care. They're de-identified, but they come from human patients. So those are classified under a different paperwork. And it's always really good to start becoming organized right when you start your project. Don't wait right at the deadline to have those documents prepared, or talk to your grant admin if you have them, to make sure that when the time comes to write your application, you have the correct forms to fill, because that can delay or get you into this very stressful situation. Christina, you have your hand up.
Yeah, and lots of times you can write the IRB protocol and even get it approved without funding. And then once you get funding, you go in, as Lauren said, and make an amendment to your protocol indicating that you've got funding. So the other thing I wanted to say, Lauren did a great job summarizing her experience. And I know this because I was kind of with her the whole time. One of the things I think is really maybe important for basic scientists to realize is you don't have to understand all this stuff. You don't have to do every step of this. You have to partner with somebody who's clinical. And that's what Lauren did. And that's what everybody that's, doing basic science, I think, should do. And that's why in CAIRIBU, we really try to put together, clinical and basic scientist researchers and build those collaborations. So Lauren didn't have to be in the hospital recruiting the subjects. I helped her, Mariana helped her, and then she did, do some control recruiting herself. But you don't have to be involved always to the same extent that Lauren was.
And thank goodness.
Anybody have any questions right now, or we can wait till we get to the end if you'd rather? Alex, fire away.
Hey, I have a quick question. For either IRBs or that may be multi-institutional. Like, so for example, say you're like, oh, I'm not getting enough samples maybe from my institution, but there's an institution close by that maybe we could partner with. I imagine that that's a lot more paperwork. And I'm just wondering if any of you have had any experience with that, or would just totally advise against that.
I have experience with that. I've done mostly human research. I've done only a couple of animal studies, but IRBs hardly ever want to cede their oversight over your study to any other institution, even if you're doing a multi-institutional thing. It's, in my opinion, really kind of ridiculous because, we're advocating collaboration and center trials and things. We know that strengthens our research, but there's no central IRB then that we would go to. So oftentimes when I've done consortium work, each IRB has to approve the study on their own. And that means sometimes in your consortium or your multi-center collaborative, some will get approval sooner than others. Some will wait a long time to get approval. That's just, unfortunately, the way that it is. And not only that, but some IRBs will request changes that other IRBs don't. And this is a problem scientifically, which I had in a consortium that we did, because all of us then had to make certain changes to accommodate the one IRB that was requesting certain things. Now, it wasn't serious in our case, and we didn't have to amend our scientific protocol too much. But what you raise, Alex, is a good point and strong reason why I think maybe eventually we should be advocating for central IRB approvals.
Yeah. Okay. Thank you. That definitely answers.
Yeah, and unfortunately, what you're saying isn't far off for IACUC either. There's no central thing, so you have to do everything twice or 3 times, depending on how it goes, how many friends you have that you're with. Okay, so we don't have any other questions right now, so we're going to move on and transition it to the IACUC. I'm just going to start with a little bit of history of it, Maria, and then you and I can get into it. So where does this thing come? The IACUC Institutional Animal Care and Use Committee. So these are a group of people that are some scientists, some not. They tried to ask my wife to be on it, and I said that was a conflict of interest. But it could be anybody to help do this. It came out of the 1966 Animal Welfare Act is where we started, where the USDA took over as a regulator of any warm-blooded animals for research, transport, exhibition, sale, etc. But the IACUC, as we know it, really came along about 1985, is where an amendment to that act from 1966, Animal Welfare Act, put the IACUC in front and center. And so, it's not, I don't know, 1985 doesn't seem that old to me, but I guess it is 40 years that we've had these. But now it's central to all of our animal research, and nobody can do animal research without having their protocols approved therein. So from my perspective, it is the most mind-numbing, but also the most important part of my job. And we can't do any of what we do without getting these approvals. And it's a complicated game. So usually the first thing you start with is a consult with a veterinarian. And that's kind of nice for us in Wisconsin and Michigan State. We have vet schools. So we have lots of veterinarians. When I was at the University of Vermont, it was a little different. There were far fewer veterinarians associated with the university, so things took a lot more time. But to start with there, just that you have your basics covered. Most of what you do, if it's rat or mouse, is not covered necessarily in the same way under the USDA as it is otherwise. As a bladder researcher, for instance, we used to use guinea pigs. Those had a whole other regulatory burden over and above what rats and mice are. So anything bigger than a rat and a mouse, the USDA animals is a whole other ball of wax where it's just, again, more paperwork. But for the smaller rodents that we're usually with, it's a matter of putting together. It's really an IRB for an animal. Is really the same thing, where you don't have surveys and informed consent, obviously, but you need to put down how many animals you're going to use, what sort of procedures they're going to receive, what surgeries you intend to do, what techniques you use to make sure that those surgeries, if they're survival surgeries, are safe, how things are fed, watered, bedded, what hours they're awake and asleep, everything in there. But most importantly is justifying the numbers. So the point of these IACUCs were to help really reduce the amount of pain and suffering of animals, but also reduce the amount of animals that we're using, so that we're not doing 160 animals just because, that we actually have justification scientifically and otherwise for how many animals we use to do all the different things that we need to do. And so, and also that there is justification that we actually need to use the animals that we say that we do. And we take that a little bit for granted, but another part of our IACUC for sure is, saying, what research have you done to show that there are not simpler models that could ask the same question, answer the same question without having to sacrifice mice or rats, etc. And so when I first started doing this, my very first one of these was in the world's most obnoxious Word document that ultimately ended up being hundreds of pages long and unbelievably redundant. Nowadays, I don't know if everywhere it does. Lauren and Maria, you gladly hop in, as we have something called Click, is what my university uses, but there's some other ones that are a digital version of your IACUC protocol. They all come with their own, again, idiosyncrasies, and each university has different rules. But by and large, those systems make it much easier to do renewals and new protocols, because once you set up your substance administration or what sort of technique you're going to use and it's approved, you can just plop it in the new one. It's almost like, cut and paste, but not really cut and paste. It's drag and drop. So this is good for the university as a whole, because they can then share protocols with you that are just standard. So I don't have to rewrite a euthanasia protocol every time. I just use the one that we use, and you plop it in. The same timeframes that Lauren talked about also apply to IACUC. It could be a couple of months. I usually start writing my IACUC protocol the minute that I know what my score is on a grant. So if I have a score that is remotely close to fundable, I start getting that going, because when the notice or the just-in-time request from Chris Mullins, who's on this call, comes to me, they usually want to show that I have IACUC approval. You want that approval letter. And so it can take a couple of months. There's triennial review. So every 3 years, you have to put it in again, which I just finished on one of mine. And you have to change things, but that's okay. The people that are going through these, it does seem like you're getting nitpicked about stuff about your research, but in the end, it's because a lot of really, really, really awful stuff used to happen. And we want to not have all that awful stuff happen again. Even philosophically speaking, even if you don't care one iota about the well-being of any of these animals and would just as soon do anything, animals that are stressed, injured, in pain, or otherwise not okay make for bad science. So even if you didn't care about the animals at all, you should care about their welfare, just because the science that comes out of it needs them to be healthy in the way that we are. When it comes to a caveat to all of this, I do social stress in mice, which is considered unrelieved stress and anxiety and pain because they attack each other. So there are justifications that you can get through your IACUC to do things that are, not, well, certain things that would never be allowed in an IRB. You can't really injure people, but you can, with scientific justification and oversight, be able to do things like that, and you shouldn't be afraid of it either, as long as you just document everything well and have a good plan for all your stuff. Maria, you have anything you want to add?
Yes, so just like Nate said, there are different platforms. Like there is another platform we use called ARIES.
Oh, yeah.
And there's also in every medical center or university you're at, there's going to be a division of animal care. And like both Lauren and Nate said, it's better to start and get to know them as soon as you can. Don't even wait for the fundable score. Don't wait for even like the review to come back. The moment you submit that R, take that vertebrate animal section and write your new IACUC. But after you meet with people from Division of Animal Care, you can ask to meet with a vet, and you can sometimes ask to meet with a coordinator or a person who's going to be in charge of assigning your application to a review panel. This is very important because you might think you're writing something as clear as sky, like spring sky, but to somebody else, it may not be clear at all. Like we perform transurethral inoculation to give mice a UTI. And it was way more easier to have a meeting with these people, explain to them exactly what we're doing. And then they were able to tell us, okay, under section C, here's what I want you to use. So don't take for granted that the way you have it written is going to be translate in the same fashion to the eyes of the reviewer. And another thing that Nate didn't mention is that if you are using biohazard agents, like for example, we're using pathogenic E. coli, Klebsiella, you have to first have approval in a different committee, right? A biosafety committee that your lab is able and approved for using agents such as those. And then those two committees, in our case, it's the Institutional Biosafety Committee. We have an approved document that says our lab is biosafety level 2. It has these pathogens, and these pathogens will now be introduced into these animals in an ABSL2 facility. If you start your IACUC with the intent of using these biohazardous agents, but you do not have approval to use them in the first place, that's going to pause your IACUC approval. And that can cause further delays. So my advice is to start early, figure out exactly what you need, and make sure that you have prior approval for all these other agents, including, let's say, if you're trying a new drug for treatment. Again, you have to have that approved, and then you have to sometimes introduce it into your protocol as a new substance, especially if it's a test substance. So all these things take up little bits of time that if you don't start early enough, it can get you to this point where like, I got my just in time and they give me 5 days to turn it around, but the IACUC committee keeps sending it back to me. So start early.
Yeah, and Maria, you brought up a good point too, that this is to be written for the lay public for the majority of it. It has to be understandable by somebody with mid-college level education is really how they tell us to go. So less is more in terms of jargon and all the other stuff that we would normally write, but it does still have to be really complete. I think one of the biggest mistakes I made on the first one I've written, because I've done that at 3 universities now, is entirely too much detail about things that happen after the animals are dead.
Yeah.
So my IACUCs historically, don't care very much about all the particulars of procedures that occur after an animal is euthanized and tissue is removed. The focus of our IACUCs have been what happens on a day-to-day basis when that animal is alive and up until the point at which that animal receives euthanasia. Now we have to justify the use of the animal in terms of why do I need to isolate a bladder? But as far as excruciating detail about calcium imaging techniques and things like that, they do not care. And that was my first one. My IACUC said, "This is nice. Would you please take out all of this?" Because it was just minutia to ask questions that need not be there. So remember, that's what is really important. From the moment that that animal is brought into your research facility and to the point in which they're humanely euthanized, that is the time period of things you need to focus in with detail. What you plan to do after it, you need to say it, but you really don't have to get nuts. I also think that IACUCs, like I said, I've done 3 of them now. Many of y'all do crossword puzzles, right? Depending on what paper you do it from, like those crossword puzzle writers kind of have their own little thing. Like I know the New York Times guy always puts a pay in there at some point. IACUCs are the same. Every single one that I've been involved with had some weird little nuance that was their thing, that everybody was really into paying attention to. Talking to other people will let you know what that thing is. But the thing also changes. That just happened to me in my 3-year renewal, is we have a new person on there. And I'm like, well, why are you talking to me about substance administration of saline? I have to add that in cystometry. But again, I just back up and think, it's true. It's the substance that goes into it. But just ask those questions about those things. At Vermont, for instance, they were very adamant that even though the law did not require it, that we split out mice by pain level. So some universities do that, and that gets really complicated because extra paperwork comes if you're pain level D, which is worse. Here, my current institution, they're like, if it's a mouse, it's C, period. We don't care what you do to it. So these little nuances, unfortunately, are things that you're just going to have to ask and get started with. But really, once you go around the horn once, you pretty much got it.
Another thing to keep in mind is that this can be done many different ways. Some people have an IACUC per RO1. Other people have one IACUC protocol, like for example, the pathogenesis of UTIs. And every time an RO1 comes in, the great hack is to say amendment January 2025, and then the reviewer's eye goes to that particular spot, and they're like, okay, everything else is the same. I'm going to focus on this amendment. At least in my institution, this has been very, very useful. And again, I wouldn't have known if I had not met with the Division of Animal Care and the review board, and they actually said, yeah, if the major thing you do is this one specific procedure, but you do it to ask XYZ questions, it's easier to have a mother protocol and then amend every time you get an R or a different kind of project initiated, unless it starts getting really complicated, in which you make an entirely new protocol, if that makes sense.
Yeah, and I'm the exact opposite because of the exact reason you just said. The 2 R1s that we have are really disparate in the experimental design. And so putting them all together in one, I could freaked. So they asked that we split them. And so it's just about that. You're exactly right. If there's a thing that you do, just put it all together. But if you have lots of different things, it can be easier. Chris, I'm sorry, I didn't see that you had your hand up.
I just wanted to echo this point about a mother protocol or umbrella protocol. It can be very helpful when you do a lot of like human subjects research, for example. And the other thing that I wanted to mention is that, and maybe Lauren could speak to this, is that, when I started doing human subjects protocols 20 some years ago, it was all by paper, and you had to go and deliver 50 copies or whatever to each reviewer. And you could give out money. We could give out money as incentives to patients and so on. Now it's totally different. And I'm looking at Lauren because we had to learn, she had to learn, we all had to learn this very complicated system that the hospital now uses. You can't just hand patients cash anymore. They've even changed it, Lauren, since you did this study. Now we have to go through even more hoops to give people their incentives. But different hospitals will have different systems. But by and large, I think they're all moving to these sort of automated sort of billing things. And you have to enter like a research order, lab order, which had to be done before the patient got themselves to the blood draw place. Lauren, that hasn't been addressed yet, and I wonder if you want to say a few words about that.
Sure, I can say a few words about that. It was pretty frustrating, especially as like, well, since I was doing the control recruitment and I needed people to get blood draws specifically, that needed to be in order through the hospital, but they couldn't be a patient of the hospital because they were controls. And it became like a really big roundabout mess just to get someone to draw blood on a person. And yeah, and as far as you had documenting all of the incentives, I thought the incentive was going to be useful because I was feared people wouldn't want to get their blood drawn for free. So you can only, it's capped at $25. That's the most you can give each individual. And it may have been more trouble than it was worth to do that. I'm not sure that I'll do it again. I might try to just not do an incentive up front and see what we get. And I would say most people that I don't think that they really cared about the money that much, to be honest. At least from the control side, they just seem to want to be part of the research. And I was actually really surprised. And that's something that I can say like a big positive, that a lot of people wanted to be a part of research, which I was surprised by. So anyway, that's kind of a roundabout way of saying that, yeah, there's lots of other little things that just make it hard that you learn as you do it. And that's just all you can do.
Cool. I guess the only thing I was going to say about these two was, I thought about this when you talked about reportable events, Lauren. IACUC is the same. So unintended or unexpected death needs to be reported in the same way. And it's really important that you do because we don't know why. Sometimes it can be an infection that wasn't caught in sentinels in the entire animal facility that led to an unexpected death. And we don't report those things. That can go on for weeks, months, years even. I have a friend that lost her entire colony in Missouri because an air conditioner failed and nobody reported that the temperature was high in the room in the summer. It was in an outdoor mobile unit while they were rebuilding. And so these sorts of things are really imperative to report. I've reported multiple of them, not again because I'm a bad researcher, because we were using some new in vivo hardware and an animal got stuck in such a way in this device that we could not free it without sacrificing it first. All of those things are important to share and know and to do. And I have, at least in my experience, have had nothing but appreciation from my IACUC and my veterinarians and my animal care staff for doing so. I've never been yelled at, reprimanded, finger wagged, or otherwise. Usually they're just amazed that we're taking time to fill out, the form and make those things. So do that for sure. The last people you want on the bad side of your existence and research is your IRB or your IACUC, followed closely therein by your contracts and grants administrator. So as long as you keep those three groups happy, go lucky, things tend to go pretty well. All right, Lauren, and Maria, do you have other things you would like to share about this stuff? And also, any questions across the board? Like I said, I've done this since I was a student all the way through as faculty. So, we can talk about all the different little bits and pieces if anybody has questions or if anybody's writing them right now.